Are T lymphocytes just cells sent back from the future to protect us? Perhaps. Last time we talked about B cells and their role in antibody production. In that I touched on a little bit about the T lymphocytes and their use as helpers. But they also have a much more important role, not only as a second signaler, but also as a terminator in their own right. Now all T lymphocytes have a T cell receptor on the membrane of their cell surface. Now this is composed of two polypeptide chains that are in many ways similar to the short arms of antibodies. The difference is the T cell receptor never binds to free-floating antigens, but only to cell surface receptors that have an antigen already bound to them. This cell surface receptor is called MHC, or major histocompatibility complex, something that I touched on in the previous section. Also, unlike other antigen-antibody interactions, the antigens that are presented and react to T cells are actually only very small polypeptides that are generated from the degradation of pathogen proteins, so not binding directly to the pathogen itself. In order for a T cell to get into the action, you have to have the antigen presented to the cell. Antigen-presenting cells and viral infected cells both will process and then digest portions of the pathogen that's either they've been exposed to or that they're infected with, and then attach it inside the cell to a major histocompatibility complex protein. It then moves the MHC molecule onto the cell surface, and at the cell surface, the MHC with the antigen attached to it is recognized by the T cell receptor. There are two classes of MHC that you need to know about. The first one is class 1, which is used to process and present intercellular pathogens, so any bacteria that's inside the cell that is infected, or especially any viruses that are inside the cell, will be attached to an MHC class 1 molecule. Class 2 MHC molecules are used for extracellular pathogens, and these are primarily used by the antigen-presenting cells to take in a portion of the pathogen's antigens, processes them, attaches them to an MHC class 2 molecule, and then puts it out onto the cell surface for recognition by T cells. T lymphocytes will only recognize one specific MHC class molecule depending on their type. So T cells called cytotoxic T cells have a co-receptor called CD8, which cooperates and binds with the T cell receptor to recognize both the cell as well as the MHC class 1 molecule. Another class of T lymphocytes are the helper T cells. These have the same T cell receptor but associate with a CD4 molecule on its surface that then allows it to recognize the MHC class 2 receptors. Often immunologists will not refer to T lymphocytes as cytotoxic T cells or helper T cells, but will instead refer to them by their CD subsite. So CD8 or CD4 refers to cytotoxic T cells or helper T cells respectively. And so in clinical practice, you'll commonly come across that naming instead of naming them as helper or cytotoxic cells. The important thing to understand is that it is the CD4 or the CD8 molecule that determines whether the T cell is either a cytotoxic or a T lymphocyte, and also is responsible for binding the specific class of MHC that's on the presenting cell. So how do we get this party started? Activating T cells, as you can imagine, takes a bit of time. And this is part of the reason why we have an innate and an immediate reaction to infections. Not only are they responsible for trying to stave off and defend against the infection, but they also are responsible for breaking down antigens from pathogens to start the presentation process. Because this involves a lot of different steps and a lot of different cytokines, this activation of T cells takes a longer time, and so this level of immunity takes in the order of days to weeks to really get ramped up. Lymphocyte activity itself actually doesn't occur at the site of infection and only occurs in the lymph nodes or in the spleen. In order for a T cell to actually know that there's something going on, it relies on a class of cells called dendritic cells, which are part of the category of antigen-presenting cells. These groups of cells capture antigens at the site of the infection from both directly engulfing and phagocyting the pathogens, or also just cleaning up some of the debris left behind by the neutrophils and the macrophages. And it takes in these antigens and then transports them to the lymphocytes for presentation. A dendritic cell will engulf and then start phagocyting a pathogen or an antigen, and then while it's traveling through the lymph system to the lymph nodes, it will continue to process it and then take the antigen, attach it to an MHC class II molecule, and present it on its surface. Once the dendritic cell gets into the lymph tissue, the dendritic cell just kind of hangs out, and T cells circulate around them and basically query the dendritic cell to see if anybody recognizes this antigen. If the T cell receptor recognizes the antigen and binds to it, the T cell is then activated, and the T cell and the dendritic cell form another pair. The T cell is now activated, and it then starts clonal expansion, producing many copies of itself, and then differentiates as well into a variety of different helper T cell subsets, which we're not going to talk about in this section. All of these different subtypes are involved in different nuances of the immune system, and will both activate macrophages, it will enhance neutrophil activation and response, and also, more importantly, go looking for B cells to start activating them. The other subset of T lymphocytes are the cytotoxic T cells, or the killer T cells. They have a very specific job to help defend the body against intracellular pathogens. You see, the issue with intracellular pathogens is that they effectively can evade all of the host defenses by simply being inside the cells, and so any portion of the immune system that encounters a cell that is infected with one of these pathogens won't recognize that the thing is infected because everything's inside and it's just one plain old regular body cell, so why would you do anything special here? And the pathogen can hide out inside those cells. Now, there's two ways that this could play out. On the one hand, the cell itself might decide it can take matters into its own hands and try to destroy the pathogen on its own, because most of our somatic cells do actually have digestive enzymes and inside lysosomes that it can engage in destroying protein-based pathogens. But that's often not the case, and it relies on the immune system to help finish off the job. So the second option that the cell has is to actually take proteins from the pathogen that's inside it, portions of it, and bind it to the MHC class I molecule and present it on its surface. This effectively acts like a flag saying, "Hi, everybody, there's something in here, something I can't get rid of. Can you come and help me?" This is where the T lymphocyte killer class comes into action. Now, a native cytotoxic T cell is activated when its T cell receptor is presented with an antigen that it recognizes that's presented specifically on the MHC class I polypeptide. Once it's activated, it starts loading up on cytotoxins in its granules as it continues to differentiate and undergo clonal expansion. Once the guns are loaded, it goes to work. And this is probably one of the cooler things about immunology that I really like. This class of T cells, now that it recognizes that there is an infection and is activated, it will go through all of the different cells that are presenting MHC class 1, and when it encounters a cell that is presenting the antigen that it recognizes, it binds to the cell. Now, unlike the T helper cells, when they bind to dendritic cells and they start co-activating and releasing all kinds of nice cytokines to help with the activation and differentiation, clonal expansion, the T killer cell kills the cell. Like a terminator, it releases a bunch of granules and kills the cell by apoptosis. And once the cell is dead, the T cell disattaches and goes looking for the next one, binding, releasing granules, killing, and moving on. Now, the beauty of using apoptosis for killing as opposed to using membrane pores to pop the cell is that it allows us to keep the pathogen contained within the cell that it was infecting. Next, after that, macrophages come along and they act as the cleanup crew, picking up all of these cellular debris and engulfing them and breaking them down so that they can cause no harm to anybody else. So in this section today we covered the cellular immune system, specifically the T lymphocytes. There are two classes of T lymphocytes that are designated by their CD4 number. So CD4 are T helper cells and CD8 are cytotoxic or killer T cells. CD4 helper cells are responsible for activating a variety of different other cell types, whereas the T killer cells are terminators that go out and take care of business.